Um, and I will, it takes a little while for it to download after we're done, but I will
Hmm.
send it to you.
Okay, perfect. Thank you.
Awesome. And we're not doing a case presentation today, so you can, we're here for an hour and a half, so you are kind of slated for like an hour of presenting and then just discussion and questions for the rest of the time after.
Hmm.
Awesome. Great.
Sounds good.
Perfect. We're just gonna give folks a few more minutes to show up. I know 8 a.m. is early, so we'll probably get started around 8:05 or so. So just hang tight, folks. Thanks for coming. Great. We're going to go ahead and get started, and I will let people trickle in as they arrive. So today we have a little bit different agenda. We're not doing a case presentation today because we have Erik Messamore here with us, and he's going to talk about cannabis use and psychosis and schizophrenia. And we feel that this is just such an important topic, and he has such valuable insight and information to give us. So we're gonna kind of give him the floor for a full hour today, roughly, and then we'll do Q&A and discussion afterwards. And I suspect there'll probably be a lot of thoughts and questions that people want to share, I hope, anyway. So thank you all for coming. Per usual, we are going to do very brief introductions so that everybody knows who each other is. And I know that we have some names I don't recognize. So I think there's some folks here today that haven't been to one of our echo clinics before. So if this is your first time here, thank you so much for joining us. And we hope that you come back. So Piper is not with us today. She is doing some trainings at the APA conference. So she sends her hellos from there. And my name is Carissa Kudre. I work with Piper in the Department of Psychiatry. I've been working with her for 5 years as a researcher, research coordinator, project manager, everything that happens that isn't something she directly does, I'm doing. So welcome. It's great to see everyone. I'm going to let Erik introduce himself next, and then he can popcorn to whoever else so we can get through introductions. Erik. Oh, we can't hear you. You're muted. There we go. Yep.
There we go. All right. Thank you, Carissa. Hello, everybody. My name is Erik Messamore. I'm a psychiatrist. I also have a PhD in pharmacology. I am an associate professor of psychiatry at Northeast Ohio Medical University, and I'm involved in a first episode psychosis program there, and also lead a schizophrenia, actually now it's a general psychiatry focused echo called SC Consult, where we talk not only about schizophrenia, but any kind of psychiatric concern that clinicians may have. So you're welcome to join me there sometime. It's on Tuesday at noon Eastern time. And then how about we go to Krista?
Hi, everyone. I'm Krista Reeder. I'm a clinical social worker, and I am an individual resiliency trainer at the mHealth Navigate program. I'll go to Orly.
Morning, everyone. I'm Orly Haven. I'm a family clinician at the Navigate program, first episode psychosis program in St. Louis Park, Minnesota. And I'll go to Kayla P.
The mute button every time. I'm Kayla Paulson. I'm the program supervisor for the Child and Family team in Duluth, Minnesota. And I am the team lead for the FEP team up here as well. I'm going to go to Cece.
My name is Cece. I'm the IRT clinician for the FEP program at the Human Development Center in Duluth. I will pass it to Alex.
Hi, I'm Alex. I am the medical student that is on this rotation this month. I can go to Kayla J.
Hello, I'm Kayla James. I'm an IRT at the Navigate program in St. Louis Park. I'll go to Andrea.
Hi, everyone. I'm Andrea Botzett. I've got a really annoying kitten here. Sorry. My apologies if she gets in. I'll kick her out soon. Anyway, I'm a licensed marriage and family therapist in the Department of Psychiatry in the Riverside office. Worked with echoes quite a bit, worked a lot with substance use over the past 25 years, especially substance use in adolescence. I think that's it. I will go to, let's see who else is on the list. Danny Jenkins.
Yeah, hi. So I'm Danny Jenkins. I'm the CISO, the Support Education and Employment Specialist for the HOPE program. And I will pass it to Nancy.
Morning, everybody. Nancy Howe. I'm the family peer for the Navigate teams at the St. Louis Park University of Minnesota Clinic, and also the HOPE program at Hennepin Healthcare. I'm also the director for the children's team at NAMI, Minnesota. And I will popcorn to Melissa Delho.
Thanks, Nancy. I'm Melissa Delho, clinical social worker by background, program director of our Navigate program with the University of Minnesota out in St. Louis Park. I will popcorn to Amy.
Good morning. My name is Amy. I am a doctoral student doing my practicum with the Navigate program out of St. Louis Park as an IRT. And Melissa, have you gone yet?
No, I lost power for a second there. So I'm Melissa Rosewall, and I'm a social worker on the Navigate team at the University of Minnesota. And I don't know who hasn't gone yet. So good morning now, everybody. Hi.
I can go. My name is Helen Newman, and I work in Cedar Rapids at a community mental health center. We have a first episode psychosis program here, and I'm one of the IRT clinicians that work for that team. So thanks for including me.
Thank you so much for coming.
I think I'm last, but hi, I'm Trisha. I'm also IRT in Grand Rapids, Michigan. So excited to be here.
Thanks for being here, Trisha. Danny, or Liz, did you just show up? Did you want to introduce yourself really quick and then we'll move on?
Absolutely. Sorry, I'm late.
Okay.
I'm Liz Green. I am the team lead in Cedar Rapids, Iowa.
Welcome.
Thank you.
Sorry, my internet just dropped out for a minute, so I disappeared briefly. My apologies. It's a great morning for that to happen. I'm glad that you didn't all get kicked out. Wonderful. Thank you guys so much. Welcome, everyone. I am going to just turn it right over to Erik for
Carissa, before we do, I think Alex, and did we get everybody else introduce themselves?
I went.
You did? Oh, sorry, I missed that. Okay, just checking.
Okay, perfect. Great. The floor is yours, Erik.
Okay, thank you.
Yeah.
Well, let me move to the share screen. And it says 2 people entered the waiting room. So Carissa, I guess you can see now who's entering the space. And can somebody nod if you can hear me? Okay, super, thank you. All right, I am going to talk today about kind of many things related to the intersecting topics of cannabis, psychosis, and schizophrenia. I got very interested in this many years ago. I've actually spent most of my clinical career in Portland, Oregon, and there is hardly a state more weed friendly than Oregon. So I got to see a lot of interesting things happen there. The thing that got me first really curious was a guy who was a man, important for the story because men in their mid-30s usually never develop new onset of something that looked like schizophrenia. But this guy did, and he had no family history of mental illness. He had nothing going on. He wasn't a patient of mine. He was an acquaintance of a patient of mine. So I was hearing all these secondhand reports. But he just loved to smoke a lot of weed, like many people do, and especially many musicians do. And then one day he just develops paranoia and and hallucinations, and stayed that way forever, as far as I know, which is just odd. And later in my career, later in my days in Oregon, I worked at a state hospital where, in the probably typical of many state hospital systems, people tend to have long-term patients, and the ones that stay a long time are the ones that don't get really great responses. And I love that job because I actually was able to learn a lot about how to treat quote unquote difficult or complex cases. And generally, I could at least get moderate but significant improvements in many of the patients that I was treating. But I noticed that every single person that no matter what I tried, I could really do nothing for, were having in common that they began using cannabis during their teen years had many years of cannabis use and then developed a thing that looked very much like schizophrenia, but seemed to not respond to treatments the way that schizophrenia typically does. So between the two sides, seeing a nuance set that is extraordinarily atypical and seeing this collection of people that had in common significant earlier cannabis use, but had nothing in traditional responses to medications, I just got really exceptionally curious. And eventually I started to talk about known scientifically credible risks of cannabis. And then I found out that when you speak in public spaces and say things like there might be some downsides to cannabis, you get attacked a lot. So I feel it's just important, since this is recorded and may go, in public spaces, that I personally think that I like liberty, I like freedom, and I like that value, like many people do. So I believe that in an open, free society that actually values liberty, that any informed adult who wishes to use cannabis should be able to do so without fear of prosecution. And I've always, incidentally, supported and signed a petition that would liberalize or reform or make more sensible or eliminate prohibition policies of cannabis. But I also think that, and this is obvious, all this is obvious, anything that is strong enough to affect mood or perception, like alcohol does, like medicine does, or that can change symptoms of illness, like any medicinal substance can, even if it's natural, is also strong enough to, strong enough to cause some risk. Medicines work, what medicines are fundamentally they are chemicals that have special shapes, and those chemical shapes allow the drugs to interact with the chemicals of life. Drugs, all times and everywhere, perturb the normal processes of living systems. When we use drugs therapeutically, we intend to perturb the system in a way that will counteract the perturbations of disease. But when you perturb a system, you always invite risk. So basically, that's a very fancy way of saying that you don't get something for nothing, or that all drugs have risk. And I've noticed that in the modern era of cannabis reform, personally, I think it came as a surprise to the reformers who had for decades made the liberty and freedom argument, that the medicinal substance argument was much more resonant with the public. So we've seen in the last 10 plus years a kind of a rebranding of cannabis as a medicine, a harmless natural medicine, and so on. But I'll tell you, it's just like any other medicine. There's no such thing as a harmless one. So I believe personally that if we're going to have legal cannabis, then we should be publicizing its risk just like we publicize risk for aspirin or cigarettes or alcohol. It's not a special drug. It's a very ordinary drug, and it can do many useful things, and it can do, therefore, many problematic things. But that's not the message that public tends to receive from many supporters within the industry. This one was a particularly great quote from a woman who was the media relations director at the National Cannabis Industry Association. Her name is Morgan Fox. She was being interviewed by the local television station in Cincinnati, Ohio, and said, there is zero proof that cannabis consumption causes psychosis. So Morgan, I actually filled in explicitly her affiliations and her disclosure information. Here's mine. I actually get $0 from anybody for talking about cannabis. I am not on the payroll of any anti-cannabis or pro-cannabis lobbying organization. I am not a minion of the state of Ohio. My views don't express the opinions of my university employer. I am simply just a guy who's been working with people with psychiatric problems. And I just got really sad and tired of seeing so many people say, I had no idea that cannabis could do that. And so I think I want to let you know, I guess I'll, respond to Morgan Fox's assertion and ask, and, go through some evidence about, can cannabis cause psychosis? Well, let's start back in the 150 years ago. We we actually had medical marijuana. It was just called medicine back in the 1800s. Back in that era, most of medicine was simply derived from natural substances. We used the raw plant material, or we used extracts of the plant material. Most of medicine was just using natural stuff, and cannabis was known has been known in Western medicine since at least the middle of the 1800s. People used cocaine, people used opium, people used lots of stuff. And there was, I think it's really informative to just put this out, that this was not a moralistic era on intoxicating plants, because medicine doctors were using them all the time. So when people were talking about risks, as they did in medical textbooks, they were not pushing a prohibition agenda. They were simply informing other doctors what you might see when you use marijuana therapeutically. And essentially, different ways of phrasing, it can cause psychosis, or it can cause significant negative effects on cognition, to put it plainly or simply. And so that was that. Then, of course, we got a whole bunch of things happened, and then the attitudes of moralism, it's always been, in my view, it's been a pendulum going back and forth through society for several centuries in the United States. The pendulum was swinging more toward moralism in the 1930s, and marijuana got prohibited and stayed in prohibition much longer than alcohol, as you know. But let's move on to the modern time. Another way we can look at answering the question, can cannabis cause psychosis, is to simply ask a bunch of people who use cannabis, have you ever had psychosis? And this is a report, a community survey done in New Zealand in 1996, I believe. Yeah, 1996. And FYI, in 1996, the THC content of herbal cannabis was probably 4% or 6%, about half or a third of what is average content these days. The simple survey asked a bunch of people, if you ever use weed, nearly 200 said that they did. And then of the ones that reported using, they asked them a bunch of questions about, have you ever had problems with X, Y, and Z? The specific question in relation to psychosis was, after taking cannabis, have you ever had strange, unpleasant experiences, such as hearing voices, or becoming convinced someone's trying to harm you, or that you're being persecuted? 15% said, yeah, that had happened with them. And you can look to a collection of similar sorts of consumer surveys. Green and Kavanaugh and colleagues in 2003 published this paper, in which they simply compiled all of these consumer survey reports that have been published, and in kind of like a meta-analysis, they simply looked at a derived large sample size, and half reported that they had experienced paranoia, 20% or 1 in 5 experienced hallucinations. So since psychosis is defined by the presence of delusions or hallucination, and since paranoia involves a belief that is probably false, that you're being endangered, then we can say that at least transient psychosis occurred in between 1 in 2 to 1 in, between 1 in 5 to 1 in 2 consumers in this large consumer survey. You can get more precise data, more scientifically valid data, by simply getting people in laboratories and giving them THC or cannabis extracts or whatever. Researchers tend not to have people smoke cannabis because it would be difficult to replicate from laboratory to laboratory, since the herbal product can vary in quality and quantity of materials. With using extracts, you can very precisely measure the amount of THC or other chemicals that are going in. And they tend to recruit people who don't have known genetic risk factors, who don't have family histories of mental illness, who don't have prior personal histories of psychiatric symptoms. And very reliably, you will find in these kinds of studies that a portion of healthy volunteers given THC or plant extracts will develop psychosis as judged by scores on things like the positive and negative symptom scale or other measures. So in fact, this review article reports on 68 studies that look at that. And we can say with some confidence that if you give people THC, some of them will develop psychosis. You can also do the gold standard and run that against placebos. You can do a large multi-site double-blind placebo-controlled clinical trial on THC versus placebo. We've actually done those because THC is a legitimate medicine that doctors can prescribe. By the way, I have prescribed it. And it goes under the brand name Marinol. It goes under the generic name dronabinol. And in order to get FDA approval, the makers of Marinol had to do regular standard clinical trials. So here is published in open access databases, the adverse effect profile. So highlighting things that happen in more than 1% or more than 1 in 100 consumers. And they got, with the asterisk here, means that it was between 3 and 10%. Paranoid reaction, hallucination happened. And that is with a oral dose of 2.5 milligrams twice a day, so 5 milligrams a day. That's far below, I mean, the average joint these days estimated to have around 12 milligrams of THC. Medical marijuana laws in almost every state that has medical marijuana actually says nothing about the concentrations or the amounts that count for that. So as a medical marijuana patient in Ohio, I could go in and I could buy a product that will get me like 100 or more milligrams easily. So anyway, the point that I'm trying to make is that this is a really low dose compared to what people will be using in today. And you got that. So, and then as an aside, since I've pointed out in public spaces like this data, then I sometimes hear people say, oh, but no, that's dronabinol, that's synthetic. And then I actually tweeted this one time. So they make this case, I call them optimists, that synthetic THC isn't the same thing as natural. One blog, Herbworthy, put up this very sort of, well, I don't know if it's deceptive or just ignorant, but they said, look, see, Marinol and THC are different. If you understand structural formulas of organic chemistry, these molecules are 100% the same. To put a fine point on it, here they've actually just drawn out hydrogen atoms in organic chemistry structural formula conventions. We don't usually draw out hydrogen atoms, so it's understood that those two H's here are actually attached to there. And the only other thing that looks different is that here they've actually written out each carbon atom, 1, 2, 3, 4, 5. Each bend represents a carbon atom. And there's hydrogen atoms there. So they actually drew out all the carbon chain, and here they just abbreviated C5H11. These are identical molecules. So this data is valid. And altogether then, I mean, any actual open-minded person needs to just concede that it's been proven by every standard of evidence, it has been proven that THC can cause psychosis. And since the cannabis that most people like to use contains THC, then THC containing cannabis products have the potential to cause psychosis in a portion of consumers. That is absolutely no longer debatable. As an aside, I'll try to walk through this quickly. As an aside, I just want to talk about a way of describing psychosis that I think is maybe nicer than some of the ways that it's described. For example, it's a break with reality, blah, blah, blah. By the way, I'm not a big fan of using the term break or broken, anything to do with human beings or their minds. So, and if you hear like parents or patients talking about psychotic break, I would encourage you to, help them to reframe that in a way that's not so like stark of a metaphor. I call psychosis the most common problem that nobody talks about. It's because about 1 in 11 people will experience some facet of psychosis at some point in their life. If you look at kids or adolescents, Here are some incidences, cases per 1,000 per year. Suicide, diabetes, teen birth. You hear all sorts of things about these things, and there are programs in place to try to minimize suicide, to prevent obesity, to prevent diabetes, to encourage better sex practices. We are actually on all of these problems that affect adolescents, and I've never seen any comparable efforts to try to promote awareness about psychosis, but that's by far a bigger risk. So again, most common problem nobody talks about. Psychosis is a symptom. I call it actually I'd say it's probably best understood as a misperception syndrome rather than a break of something. The brain, psychosis happens because the thinking parts of the brain are trying to make sense of clusters of misperceptions that the perceiving parts of the brain generate. And when those misperception clusters occur in areas of the brain that produce sensations, those give rise to hallucinations. And when the brain starts to misperceive the importance of things, then the thinking brain has to make sense of why does this and this seem like they are almost connected. And that's how psychosis happens. And so that's that. Now that we've established, as an aside, a way of thinking about psychosis, and that we've established that THC absolutely can induce psychosis in some people, then the next question is, how can that happen? And it happens, well, I'll run you through some background information and I'll explain. I think I'm going to talk about 5 possibilities that make some sense from a scientific point of view. But fundamentally, why marijuana is more interesting to human beings than broccoli is because marijuana contains this molecule called THC, tetrahydrocannabinol. And here they've drawn that structure of THC, but they've actually put some other, sort of how a cell might see it. It's this kind of cloud of chemical influence. And it looks relatively similar to this chemical called anandamide. Anandamide is a compound that brain cells make to communicate with each other and to regulate the exchange of information. And THC looks like an anandamide, so it kind of tricks cells into thinking that they're getting anandamide signals. It behaves like anandamide in that fashion. The significant difference, though, is that the body has processes to create anandamide on demand. And once the anandamide has served its purpose, it has proteins, technically called enzymes, excuse me. It has these enzymes which are designed to very rapidly get rid of anandamide. Because the thing with signaling molecules is they need to be brief. I'm going to mute while I do this. I'm getting over COVID, so hopefully I can get through this. Okay, so the thing with signals is that they need to be brief or pulsed. So if the body makes an anandamide and it floats around for 5 minutes, it is useless. The body has to destroy it almost instantly. THC is not affected by those destructive enzymes. So with THC, you can achieve massive concentrations of signal, and the body has no way to break it down rapidly like it does with anandamide. So that gets you to some points I'm going to make later on. But that's basically THC and anandamide are very similar. Anandamide binds to a specific kind of protein. It's like an antenna. We give it this weird name called CB1 receptor. And THC, because it's shaped like an anandamide, can also attach to that CB1 receptor. Receptors are, I mean, they're kind of very much like antennas. They are on the outside of cells, and they can detect chemicals in the outer layer or outside world. And then they can send the signal inside the cell to let the inside of the cell know that the outside signal is there. The CB1 receptor is located in lots and lots of places, and I'm not gonna, there's not gonna be a quiz on this, but I point out all these different brain regions just to make the point that it's a very widespread signaling system. And the general rule is that the more places that you have that respond to a drug, then either the more the greater the number of possible benefits, or the greater the number of possible risks. So that's just how it works. I want to point out, though, cerebral cortex is involved in decision making and in perceiving things. Hypothalamus is involved in the regulation of stress. I'm not going to talk a lot about this, but data from long-term cannabis exposure suggests that, let me put it this way. Drugs are chemicals, as I said, that perturb biological functions. When the body is exposed to a drug on a chronic basis, the body acts as if it's under chemical pressure. And so the body responds very well by changing its function in a way to oppose the chemical pressure. So that's the process of tolerance. And data suggests that because the THC can impact stress processes in the hypothalamus during the process of tolerance or fighting back against chemical pressure, the set point for stress changes. And there's a lot, paradoxically, long-term cannabis users have, in some cases, evidence of severely dysregulated stress response systems. The amygdala is an area which generates the perception of fear or threat. Hippocampus is for memory, and we'll leave it at that for now. But main point, it does a lot of stuff. So it has, it has potential for a lot of therapeutic benefits. It also has potential for a lot of negative aspects. Let's talk about this other thing that as you might imagine, I read a lot of the writings of people who are on the pro-cannabis side, and people tend to take, I don't know what the right word is. They tend, when they explain about how cannabis works, they are relatively limited in the extent of their explanation. They'll say, as I said, it looks like an anandamide. It activates the CB1 system, the cannabinoid receptors as part of this thing called an endocannabinoid system, and it's related to blah, blah, blah. So they kind of leave it at the first layer of chemical interactions. But let me take you through the next step, what happens immediately after the anandamide comes out. So this is a model of a synapse. Here you have a nerve cell that is sending electrical signals. Then it ends. So it has to, it ends. It has to bridge the gap. It can't send, it's not just a continuous wire. So it has to convert the electrical signal into a chemical signal. So these little round dots represent some kind of neurotransmitter. And then the chemical diffuses across this narrow space and attaches to those special receptor proteins, which detect that something happened. And then they turn on processes inside the receiving cell, and then can either make the receiving cell more likely to send electrical signals or less likely to send electrical signals. That all depends upon what the transmitter is and what the receptors are that are detecting it. I mean, whether it goes active or becomes more active or less active depends upon what the nature of the chemical and the nature of the receptor. But in many, many cases, almost immediately after this receiving cell gets the chemical message, it produces anandamide. So that's represented by these hexagons here. Once it says message received, it creates anandamide to float back to the input cell to say, basically, message received, I got it. So in that case, then the incoming cell doesn't need to keep sending the same message. So anandamide functions as a way to say, I got your message. And these anandamide molecules will attach to that CB1 receptor, and when the CB1 receptor is activated by anandamide or by THC, it basically tells incoming cell, we don't need to send out so much chemical anymore. Message received, mission accomplished. So the ultimate or the next step after anandamide binds to that CB1 receptor is it essentially temporarily turns off the incoming activity. And that's why it has all the effects that it does. So many, many, many neurotransmitters are regulated by that system. It decreases the release of glutamate, of GABA, it actually, in short term, it appears to increase dopamine release. In long-term use, it decreases dopamine release, and data suggests that long-term use is associated with suppressing the production and release of serotonin as well. Let's talk about each of these things now individually. Dopamine, I think since you are an audience of people who work with people with psychosis, you know that the leading theory of psychosis is that it's related to excessive production of dopamine. We know this because drugs that are pro-dopamine can reliably cause psychosis as a side effect. And we know that drugs that block dopamine, i.e. most of the antipsychotic drugs, can be relatively reliable at relieving psychosis. So excess dopamine is well known to be a psychosis neurochemical cause. And cannabis, they've actually shown this in PET scan, living human beings. It's been shown that the short-term use of cannabis causes overproduction and excessive release of dopamine. There's the science, if you want to go look it up, so you know I'm just not making up stuff to scare people. And but although I'm not, like, as I said, I'm not a prohibitionist, but I do think people should understand that THC crosses the placenta, crosses the fetal brain, gets into the fetal brain, and can, actually affect brain development. So in so many ways, I want to just, since I'm talking about dopamine, point out that in rats, in rat mothers that are given THC, their pups wind up as fetuses with dysregulated dopamine systems. Their dopamine system is more active. There are sorry for the if this is offensive to some people, but there have been studies of aborted fetuses that were done in Sweden, I believe, and they were looking at the dopamine genes in fetuses of mothers that were using cannabis and compared them to not. And it seems like the same thing in animals is present in the developing human brain. And that might be related. There's a small number of studies that are following kids that had in utero cannabis exposure. And those studies are finding that it's affected with like some neuropsychological abnormalities during development. Among them, there is a kind of quiz that you can use to assess thinking style, and kids whose mothers were using cannabis during pregnancy tend to have a more psychosis-like thinking style compared to others. So there are, whether this one is related to the dopamine, nobody knows, but I just want to make the point that one possible mechanism for THC to cause psychosis is by messing with the dopamine system. The chemical interaction that probably, in my opinion, is the most significant is with glutamate. Glutamate is a neurotransmitter. It's also an amino acid. It's also used as a kind of flavoring agent, a flavor enhancer, in monosodium glutamate. So glutamate does a lot of jobs, but in the brain, nerve cells make it and they use it as a neurotransmitter. And in fact, glutamate is, by number of chemical connections that use it, glutamate is the number one neurotransmitter. Half or more of brain cells utilize glutamate as their signaling molecule. Glutamate is almost always, no, I think it is, I can't think of any example where it's not. Glutamate is an excitatory signal. So when glutamate is released, it will make the receiving cell more active, and it works pretty fast. So places like the cerebral cortex, where you generate those initial sensory perceptions, use glutamate. They use a lot of glutamate in those sort of neurological computations that generate perception. It's also massively important in a different sort of glutamate process is very strongly related to the formation of memory. And drugs like PCP, drugs like ketamine, drugs like dextromethorphan, all of them are drugs that basically block some of the receptors for glutamate. So we know that decreasing the strength of glutamate signal is a way to mess with perception, mess with memory, and a way to create a syndrome that looks very much like schizophrenia, psychosis. And again, the primary effect, I mean, the most notable neurochemical effect of THC overall is to decrease the production and decrease the release of dopamine. Why I say it's the most notable effect is because most of the brain uses glutamate, and glutamate cells have those CB1 receptors in many locations. So it's like taking the foot off the gas pedal. Really amazing study was done published 2019. They used a imaging technique called magnetic resonance spectroscopy. It's a little bit like MRI, but it allows you to measure the concentration of chemicals in a brain. One of the chemicals you can measure is a metabolite of glutamate. And similar to many human laboratory studies that I spoke of earlier, they got a bunch of volunteers with no prior psychiatric history, no family history of mental illness, healthy volunteers. They gave them THC, and a portion developed psychosis. That's all like not surprising because that's been done many times before. But what was really interesting is that they had them in this magnetic resonance scanner before and after, and they found in the people that developed psychosis, they tended to have, you could tell who developed psychosis based upon their glutamate activity. So people that were going to get psychosis had glutamate signals that suggested that they were like their gas pedal was really high, that they were using a lot of glutamate. They had a very active glutamate system. And when they were developing psychosis, then the signal changed in ways that suggested that they were having the biggest buildup of glutamate because they could no longer release it. So essentially, this study, although small and needing replication, would suggest that the subset of people who can develop psychosis from THC are the ones who are on the higher end of the glutamate use spectrum when their brains are operating. So there you go. That's a great mechanism. GABA is like the opposite of glutamate. GABA is another, technically it's an amino acid. It's the number 2 neurotransmitter by volume. 30 to 40% of brain cells use it to communicate. It's like the brake. And just as THC causes the inhibition of the release of glutamate, THC also inhibits the release of GABA. So it's like taking the foot off the brake. Science suggests that that's very important in the side effect of anxiety from cannabis, because that amygdala, which has a lot of those CB1 receptors, is extremely important in assigning the perception of threat to stimuli. And amygdala, there's a lot of GABA in the amygdala. So if a person has an amygdala that is like under a lot of GABA control, then taking the foot off of that brake means that their amygdala will start sending alarm signals up to the thinking centers and down to the body reaction centers. And in imaging studies, where they tried to look at the density of those CB1 receptors, it turns out that the subset of people that got anxious in response to THC were the ones that had the higher density of CB1 receptors. So their amygdala was able to attract more of the THC, and that seems to be the more THC got in there, then the higher the risk of having a anxiety or panic reaction. So that's a GABA effect. Serotonin, we tend to think of as a pleasant mood molecule, but there's a receptor, protein receptor called 2A. Oh, before I get to that point, I want to talk about some mood studies. Animal studies, when you give rats THC on long-term basis, it looks like they don't make serotonin very much. There's a study that was done in rats that looked at the effect of the antidepressant citalopram, which is a serotonin reuptake inhibitor. And when you gave rats the citalopram, you put a little probe in their brain to measure serotonin, and the serotonin level shot up like 400% over baseline. But in the rats that had been given THC over a long-term basis, citalopram did almost nothing to boost the serotonin levels. That's probably because they're not making very much serotonin, so there's less serotonin to be reuptaken. So there's that. And then in a study of medical marijuana users who were using an app called Strain Print, it was found that with short-term use, they had an improvement of mood, but mood deteriorated over the course of like a year of observation. So it's not, maybe not the best thing for at least some people in terms of mood. And as a tangent to this, there are a number of associations. I mean, a number of studies are linking the use of cannabis to an increased risk of suicide. And if that is a causal relationship, then it may hinge around the disruption of serotonin signaling. But serotonin also, there's a type of receptor called 2A, and the 2A receptor is like a primary target for hallucinogenic drugs like LSD or psilocybin. And a study in rats, in adolescent rats given daily THC, found that regular exposure to THC during adolescence produced 2A receptors, which were substantially increased in number, substantially overreactive to serotonin. The authors used the phrase pro-hallucinogenic conformation. So that might be a way whereby long-term cannabis use can produce a vulnerability to psychosis, which is also interesting. I'll just go, since I'm kind of running out of time, I'll just jump to this topic. The possibility that chronic cannabis use produces a serotonin-based vulnerability to psychosis is very interesting, because that would suggest that dopamine blocking drugs might not be so good if the psychosis is a serotonin syndrome or a serotonin sensitivity issue. Similarly, if the psychosis or the schizophrenia-like symptom that may evolve from THC turns out to be a glutamate disruption, then antipsychotic drugs, which are dopamine blockers, are going to be minimally interacting with this system as well. So the fact that THC can affect these systems that are not dopamine in a way that might make psychosis more likely sets up the possibility that the cannabis-associated psychosis is not going to be responsive to medicines the way that ordinary psychosis is. And finally, anandamide, which I talked about earlier, that thing that the brain makes, the brain cannabinoid, it appears to have some neuroprotective and anti-inflammatory benefits. It also looks like giving plant cannabinoid will tell the brain it doesn't need to make its own cannabinoid. So some studies suggest that external THC can cause the brain to downregulate its own cannabinoid production. And that sets up this interesting concern. This is speculative, but I think it's worth, nobody's like said yay or nay whether this is a valid idea or not, because it's simply not been tested. But it does set up the concern that if THC is not as protective as anandamide and you're making less anandamide, then that person may have more vulnerability to inflammatory processes. And inflammation is another pathway to psychosis or to enduring schizophrenia-like psychosis. And it does look like anandamide levels correlate with the severity of psychosis symptoms in some people. There's really, really, really not a lot of data on this. And this last one is pretty speculative, but again, I think it's just worth knowing about. So to summarize, there's a lot of overlap between the pharmacology of THC and what we know of the biochemistry of psychosis. We know that glutamate disruption, serotonin disruption, and dopamine abnormalities are all related to psychosis occurrence and risk, and THC can knock on all these doors in addition to doing something maybe concerning with anandamide. Let's move. I'm going to try to go, I'm going to go probably over the allotted time, but I'll try to finish in time for questions. Cannabis and schizophrenia. So psychosis is a symptom that has a lot of causes. One of the things that psychosis is associated with is schizophrenia, which is not really an explanation. Schizophrenia does not explain psychosis. Schizophrenia is just a way of labeling psychosis that cannot be explained by other things. So psychosis, schizophrenia is defined as a long-term or chronic condition in which psychosis is a prominent, persisting, or recurrent symptom that is not better explained by medical explanation, or that is not part of some other psychiatric syndrome. So essentially what the DSM says is we're going to label schizophrenia any form of persisting psychosis that we can't give some other label to. So schizophrenia technically is like this wastebasket diagnosis of idiopathic or unexplainable psychosis. And I personally think that one of the biggest hindrances to progress in schizophrenia is that it's that this name does not end with S. So we tend to think of schizophrenia as a single illness with maybe a single cause and a single class. Antipsychotic medications should work. But if schizophrenia is in fact 12 different diseases that produce a similar top level picture, then that explains treatment resistance very well. So schizophrenia, that's what's good. I just want to mention that because treatment resistance, well, I'm sure you see it a lot. And usually when drugs don't work, it's because the thing that the door the drug knocks on is not the door that the symptoms are living in. So, so there's that. Let's move on to the relationship between cannabis use and schizophrenia. This, again, unbiased, not on anybody's payroll, National Academy of Sciences and Engineering, were commissioned by many stakeholders, including many legal marijuana states, to do a comprehensive, thorough scientific review of the health effects of cannabis and cannabinoids. You can get this for free online at, like, just look up National Academy's cannabis report and you'll find it in Google search engine, but free. It's big. And they conclude substantial evidence of statistical association between cannabis use and development of schizophrenia, with the highest risk amongst most frequent users. So that's basically saying that people who use cannabis are at higher risk of developing schizophrenia or schizophrenia-like symptom picture, and that the risk of schizophrenia is associated with increased use. There are actually, I think this is a meta-analysis looking at 10 different studies, epidemiologic studies, that look at the degree of cannabis exposure in earlier life and the likelihood of a schizophrenia-like diagnosis later in life. And you'll see two things that I think are worthwhile. The greater the degree of cannabis exposure, the higher the degree of schizophrenia diagnosis risk goes. And that when you see a relationship between dose or exposure and effect, that's consistent. Doesn't necessarily prove, but it's consistent with the possibility of a causal role. If you summarize, or if you make an average of all of these odds ratio, which means like the likelihood of occurrence in cannabis users compared to the likelihood of occurrence of not. So it's basically like your odds. So if you take the average of these odds increases, you'll find it's about 3.7. So we'll say close to fourfold increase in the likelihood of a schizophrenia diagnosis in regular cannabis users. So critics will say that, okay, well, yeah, there's an association. I mean, you can't deny there's an association, but they'll say it's not a cause, or that the only thing this proves is that people who are going to get schizophrenia already have genetic risk for schizophrenia. That they'll basically say weed doesn't cause schizophrenia; it only like activates underlying genetic risk of people who people who would ordinarily get schizophrenia anyway. But let's look at that for a second. So here's a great study from Dunedin in New Zealand. It's very unique. They followed about 1,000 kids born during a single year in a single geographic area. And they just tested them, measured them, extensively evaluated, collected reams of data on them every year, every 3 years, and they're still being studied. And one of the questions that was asked at age 15, have you used cannabis? And those that said yes, but 2 times or less, or not at all, they had a risk of, at age 26, of being diagnosed with a schizophrenia-like spectrum condition of about 3%. Those who at age 15 reported using cannabis more than 3 times had a 10% risk of being diagnosed with schizophrenia at age 26. Again, you can say, you know, genetics, you're only looking at genetic risk maybe. But this other study of that cohort from Dunedin, they looked at a mutations in the gene COMT, which metabolizes dopamine. And there are, we'll say, 3 flavors of the gene, or 3 ways the gene can be configured. One is with both chromosomes having methionine. Let's see how to do this simply. We'll say there's 3 types of genetic variation in this gene. One called MET-MET, one called VAL-MET, one called VAL-VAL. And you'll see that here in the people who had been not used cannabis, and those who had used cannabis, so gray and black, there was really no effect of cannabis use in adolescence on the likelihood of schizophrenia diagnosis at age 26. If you look at the people with the COMT gene configuration of VAL-MET, you'll see that those who use cannabis at age 50, in adolescence, had maybe at least a numerically higher risk of developing a schizophrenia diagnosis compared to those who didn't. But if you consider the variability, it might not be that significant of a deal. But here, if you look at the people that had this VAL-VAL configuration of the COMT gene, you'll find that those that use in adolescence had a much lower risk of developing schizophrenia diagnosis compared to these who did. And so essentially what this is saying is that carriers that basically the VAL form, and hardest in the VAL-VAL form, means that it can mediate an association between adolescent cannabis use and later life development of schizophrenia. So yeah, cannabis use is a problem for people that have genetic risk, maybe, but then the question is, what portion of the population has this particular gene configuration? Well, the authors gave that answer. 25%, and 50% carry 1 dose of the VAL, and 25% contain the high risk association from the study. So it's complicated, but you can say, to go back to this, just because you have genetic risk for schizophrenia doesn't mean you're going to get schizophrenia. If I have a twin brother with schizophrenia, an identical twin, and I share 100% of his DNA, my schizophrenia risk is only 50%, which means that environment has a role to play, and cannabis is a factor in the environment that can activate genetic risk, which might not be otherwise activated. So to summarize that. And then they'll say, yeah, but these are all associations you can't prove. You just can't prove, this association doesn't mean anything, show me cause and effect. And then to that, I say, you cannot show cause and effect when it comes to a harmful effect. So if we want to prove that cigarettes cause cancer, we don't get 20,000 teenagers and have 10,000 become smokers and 10,000 not, or placebo smokers, and then follow them and see how many new cases develop. You cannot do this experiment. And they didn't do this experiment to establish that cigarettes cause cancer. What you do is you say, is it biochemically plausible that cigarettes could cause cancer, or that cannabis can cause schizophrenia? Yeah. Can we model this in animals? Can we give rats cancer by giving them tobacco, or can we give rats something, behavioral changes or physiological changes that look like schizophrenia by giving them weed? Yes. In human volunteers, can we create it in the laboratory in a small degree? Yeah. Is there a relationship between the degree of exposure and the likelihood of schizophrenia risk? Yeah. And so, I mean, you can never do the definitive placebo-controlled trial, and critics will always try to say, use something. They won't say this exactly, but this is essentially what they're saying when it's not been proven. But in terms of everything else, you can't get any clearer of a signal to say that cannabis use probably does create in some people a schizophrenia-like condition that would not have been there were it not for the cannabis use. And so that's that. Most people who develop a cannabis-induced psychosis, it's transitory. It lasts a few hours or a couple of days. But then what's the significance of having a single episode of cannabis-induced psychosis or THC-induced psychosis? Well, studies from largely done in Scandinavia say that basically it proves that you have a combination of brain and genes which is capable of experiencing psychosis. And the cannabis has now shown you that you have that configuration of brain wiring and genes that make psychosis possible. That might explain why in Finland, amongst 18,000 people with a single episode of substance-induced psychosis, 46% wound up with a schizophrenia spectrum diagnosis 8 years later, or 47% in a Danish study looking at nearly 6,800 people with a single episode of cannabis-induced psychosis were diagnosed with schizophrenia or bipolar disorder years down the road. So basically, having a single episode of cannabis-induced psychosis may be one's last best chance to avoid having this. Certainly, as I said, it says that this person's brain-body system is at high risk of developing a more serious persistent psychiatric condition. And this is nothing. I mean, we have almost no data to say if I have a single episode of cannabis-induced psychosis and I stop forever, will I like negate my risk of developing bipolar or schizophrenia later on? We don't really know very well, except for 57 cases that were studied, of which 35 could be followed up. And it seems like those who stop didn't have a recurrence, and those who resumed did progress to worse symptoms later on. So that's what we know about the significance of a cannabis-induced psychosis. And then, what about the syndrome of somebody who uses cannabis, develops psychosis, and the psychosis doesn't go away, or even if they stop using cannabis? That's been described in at least case reports in literature. And I've seen this in my practice, and you've probably seen it in yours. And many of my psychiatric colleagues have noticed this as well. There seems to be two things. There can be people who use a lot of weed, maybe stop and develop schizophrenia, or people who use a lot of weed, sometimes use a little bit of weed, develop something that looks like schizophrenia, and then it just persists. I'm calling this cannabis-associated persisting psychosis, even though it looks a lot like schizophrenia in most instances. And what to make of that? Well, we know that from a study in UK, cannabis use in first episode patients was associated with more hospitalizations, longer lengths of stay, and notably higher rates of medication switching or polypharmacy, which are what psychiatrists do when patients aren't getting better with medicine. So basically, this is a good marker for worse illness and higher rates of treatment, of quote unquote treatment resistance. And since most antipsychotic drugs are dopamine blocking, when people don't get better with dopamine blocking drugs, that suggests that their form of schizophrenia might not be a dopamine psychosis. And this is an area where, I mean, it's an extremely important area. It's just really hardly been studied at all. So we're left with like a lot of clinical experience, anecdote, and really hardly any hard data from, this is one of the best studies that we have. And it's, good, but not enough to make, super solid conclusions. I do want to leave this with you, though. I mean, so I'll say, in my experience, this kind of cannabis-initiated persisting psychosis that can persist even when the weed is gone is really, really, really difficult to get a person better with standard antipsychotic drugs. That's been the experience of psychiatrists that I speak with, and it's been a little bit reported, in some handful of studies in the literature. There are, though, just to note, two case reports, which between the two of them describe 3 people, but in one of them by, I think it's a German group, Levecki and colleagues, or they may be in Switzerland, I can't remember. But anyway, from European group, they described 2 men, I believe they were men, and both of them had used cannabis and developed this cannabis-related persisting psychosis. Both had multiple failures of antipsychotic drugs. In the case report, both of them were switched to a drug available in Europe called Parazine, which is very similar to Thorazine here. And and they still didn't get better with that medicine. But to that, they added carbamazepine, aka Tegretol, and noted that whereas these people had no response to multiple trials prior to this, within a week, in both cases, the Tegretol started to get them significantly better. In one case, they described the man as being able to return to school. The other one, they didn't really provide a lot of longer-term follow-up data on. And then this report by Pereira and colleagues describe a young man, 20 years old, who basically developed intense paranoia that persisted for some period of time after a single use of cannabis. And this person had no personal prior history of illness or family history of illness. The person used cannabis like a month or two later, and developed a worse paranoia that persisted even longer and got to a level that required hospitalization. And from there, he was given a variety of antipsychotic drugs, which seemed actually to have made him worse, because he went from being severely paranoid to a level requiring hospitalization to, with antipsychotic drugs, having visual, auditory, and olfactory hallucinations. That led them to try Depakote because they wondered whether they had caused some sort of temporal lobe epilepsy thing. And Depakote worked. It was remarkably effective, whereas nothing else had. They eventually stopped the antipsychotic drugs, added lamotrigine because he later became somewhat depressed, and he then was able to go back to school. I infer he came very close to reestablishing his baseline level of functioning with just Depakote, which eventually was anyway. Point is that anticonvulsants worked in these cases, and I think that's just interesting to know and maybe useful when you think about caring for some of your patients in the future. Also, in looking at this literature, that one case report, Pereira, their patient appeared to have gotten significantly worse because of antipsychotic drugs. As I said, new onset of hallucinations in multiple modalities. And another report of somebody, this was a woman, also I think in her 20s, who developed, she had a more complicated medical history, but she seemed to have a cannabis-associated persisting psychosis. And when they gave her olanzapine, she got even worse. And you can invite me for another time if you want, and I can tell you about like 40 or 50 case reports suggesting that second-generation drugs can worsen psychosis or cause mania. It's maybe very rare, but it's not something we look at since we're looking at it. And since we're not looking at it, you really can't know how common it is. But I guess, it's possible. And these two case reports, noted that. So there's that, I think, worthwhile to know. And finally, to summarize, if you have a person with a cannabis-induced psychosis, it means that they have the ability to have psychosis. So is associated with higher risk of later developing a diagnosis of schizophrenia or bipolar disorder. So I would counsel anybody who's had that to understand that they have had now a clear-cut demonstration that they have genetic or physiological vulnerability. So they maybe would do well to not mess with that anymore. And to maybe focus on, building up some other resilience factors. And we just don't know enough, but what we think we know so far would say that ordinary antipsychotic drugs don't work as well as they do in ordinary schizophrenia in somebody that has a schizophrenia-like picture in which there was a significant amount of preceding cannabis use. And if that's the case, well, actually, in any case, if you've tried a dopamine blocking treatment strategy and a person has not gotten better, and you try another dopamine blocking treatment strategy and the person doesn't get better, then probably this means that their symptoms don't have much to do with dopamine, and it's worthwhile to think about treatments that involve other mechanisms of action. So that's just a general guide I have for treatment-resistant thinking. And so cannabis can cause psychosis. It may be up to 1 in 5 people can experience it in the short term. Increases the risk of schizophrenia diagnosis by a factor of 4. And the idea that cannabis can cause schizophrenia is biochemically plausible, consistent with animal studies, proven in human labs, and consistent with population. We ought to be widely publicizing this risk information, as we have so many states that are calling it a medicine. This is about 2.5 years old, so it's probably different. But these are all the states at that time that had medical marijuana laws. Here's the number of medical benefits that were implied by their medical marijuana policies. And in some cases, you had like 30. Illinois, when it was medical marijuana, only had 40. Oklahoma actually said any condition that any doctor thinks might benefit, so potentially infinite. So the large number of benefits are being like endorsed by state governments. On state government websites, these are the number of risks that are disclosed to consumers on the public facing information. Congratulations to Minnesota for at least listing 5 of them. And so I talk about this because, like I said, I get tired of people learning it, get sad and tired of seeing people learn about it the hard way. Currently, a lot of people think it's actually good for you. And half of the adults say no possibility of potentially serious things. 2/3 of kids are saying no possibility that it's going to be difficult for me. And, you know, it works for many people. And if it works for somebody, I think that's great, but I think we need to know that it's not for everybody. And so there's that. So sorry I took longer than planned, but there was a lot. Hopefully it was worthwhile. So I'm going to stop this and I'll be around for 14 more minutes.
Thank you so much. I'm sure that there's questions, so I just want to open the floor to anyone that has anything they would like to ask.
I have a quick question, or maybe more of an observation. That's just the fact that this last month, Minnesota legalized low-dose edible cannabis products as long as they were hemp derived. And that's something that I haven't fully wrapped my head around how that's going to impact our patients. And while people can only get 5 milligrams or less of these edible products, they can also buy a whole bunch of 5 milligram edible products. So I think it is likely going to start to impact the Minnesota folks that we treat. And I know not everybody here is from Minnesota, but just throwing that out there.
Yeah, edibles are probably worse in terms of hazard. There are a couple of reasons why there are some reasons to be concerned. First of all, smoking is just, if you don't smoke, you're likely to find it nasty. And, you know, cannabis doesn't really smell that good. So if you're completely naive, the idea of lighting up and smoking can be off-putting enough that you're not going to try. But a gummy bear, you can, it's a different, like it lowers the bar for experimentation. So it can make it easier for curious people who would have otherwise crossed that to get introduced to it. What is well described in anecdote and case report is a phenomenon where I take my first dose, I wait for an hour, nothing's happening, so I take another one, and then maybe I get a little bit of a something, so I take another one. And the problem is that between the absorption takes, digestion absorption takes a while, and you get this gradual rise of blood levels, which doesn't get you subjectively high or get a subjective change. The biggest perception of consciousness changing for almost any drug is when the levels are rapidly rising. So in the case of alcohol, for example, if I drink, like 3 shots within 15 minutes, I will feel very drunk very rapidly because my levels are going very high, very fast. And I can be, as I go from, 0.03 to 0.12, I am feeling extremely inebriated. But I get my peak, 2 hours later, I'm coming down, 3 hours later, as my level goes from higher through 0.12, I'm probably actually can safely drive a car. I'm certainly not subjectively impaired. I don't feel buzzed, and I objectively can have better psychomotor performance than I did when I was at the beginning of my intoxication. So the levels going down. I mean, the best high is when levels are rising, I guess, and when they rise rapidly. So if you wind up with a gradual change of THC levels as it leaves the gut and leaks into the blood, you don't perceive it as a strong effect. And people can then chase that with adding more because, well, my first gummy didn't do anything, so I'll try another one. But then 3 hours later, you wind up with this very high level of THC that impairs many, many things. And so you wind up with a lot of most hospitalizations for psychiatric adverse effects come from edible use. And that's because people don't have an ability to gauge how much they've gotten into their system. In contrast, when you inhale something, it goes, I mean, it's very ingesting, it has to go to the stomach, it has to go through the intestine, it has to go through the liver, and then it has to go to the blood, and that takes slow. Inhale, within a second, it's in my blood, it's in my lungs. Lungs are receiving all the blood vessels in the lungs go immediately to the heart, and from there right up to the brain. So within seconds, you start to equilibrate lung concentration of chemical with brain concentration of chemical. And so if I take, one hit or two hits of a vaping or smoking device, I start to feel the effect within minutes. And so then I can understand, okay, that's enough for me. I'm really high now. And then I put it down. So consumers can better gauge their levels through inhalation routes. So edibles, probably, I mean, they're probably very useful for some people, probably more as a medicinal thing than others, but they come usually with a higher risk.
I'd be interested to see the research in a couple of years after, these vapes and cartridges are very popular with young people that I work with, and they have such higher levels of concentration that I would assume that a lot of these numbers are going to rise really quickly. I got a question on, I think it was slide 48. I don't know if you have to go back, but this was about like substance induced, a first episode. And that I think you said 26% or 46% had gone on to develop a schizophrenia spectrum disorder. Were those folks who had continued to use cannabis and THC?
I don't think that they were able to capture it very well. These were epidemiological studies. I mean, the great thing about Scandinavia is that it's a single record system. So you can very easily identify all cases of substance-induced psychosis in your country. And then you can easily identify who's got a diagnosis of whatever at whatever time point you want to do the observation. So they set up the designs to look for those two things. It's huge labor to try to go through all the records and understand who's using what and what happens between initial and follow-up. Most likely, most of them are continuing to use because I say that because in my experience, most people who have a substance-induced psychosis eventually go back to using the substance because that's just the way people are.
Thank you.
Additional questions?
You might have addressed this in the first few slides. I missed that, so I apologize for that. But my sense of the literature is that they're moving in a direction of trying to separate THC from CBD and think about, okay, which one is showing benefits here for specific medical conditions. I mean, it's not as relevant for our, I think, young people using weed, that they can't buy something that's pure CBD and hopefully not pure THC. But do you have a sense that the literature is going to become, better at saying, okay, THC is the reason for these negative side effects, and perhaps people with this genetic predisposition can use CBD products or hemp products that don't have high concentrations of THC?
Yeah, I think theoretically, as we make cannabis more legal and easier to study, we should get some better data. There are a lot of individuals or entities that are very much interested in exploiting cannabinoids for therapeutic benefits. now that I'm talking out loud about this, I think the most likely path forward is going to be that pharmaceutical companies are going to make derivatives of various cannabinoids, and they can patent them, and then they can test them, in a traditional way of testing medicine, and come up hopefully with good solutions for maybe preventing Parkinson's disease or whatever else. So that's probably how it's going to go. But ideally in the science world, if it becomes easier to study cannabinoids, then we'll see more researchers that are trying to look at defined concentrations or ratios of things and ask more specific, questions about, say, hypothesis-driven questions about whether THC or CBD or a certain ratio is helpful. And so we should get some better research on that. So far as what we see, it looks like most of the negative effects are attributable to THC. The CBD seems to not have a lot going on. CBD has actually been tested in the form of a drug called Epidiolex. It's been given double-blind placebo-controlled studies. It has not a bad side effect profile at very high doses. I mean, doses that a consumer can't really afford over the long term. There, with the exception that there's like, I don't know if it's like 4% or 3% agitation or something that you can look at as a negative psychiatric event, happens in a minority of people, but like 4 or 5 times higher than the placebo group. So a small fraction of people might have some sort of psychiatric adverse reaction to high doses of CBD. And high doses of CBD seem to have some risk for liver function. But other than that, the side effect profile of pharmacologically dosed CBD looks pretty good. And in the realm of schizophrenia or psychosis research, there have been really three clinical studies of some significance looking at CBD as a treatment for schizophrenia-like psychosis. One of them was a failure. One of them was like technically successful, but clinically was probably meaningless. And one of them, the one I think is most interesting, was done by that Swiss or German group led by Marcus Levecki. And Levecki looked at CBD in people with schizophrenia. Levecki measured anandamide levels in the blood pre and during treatment. And he found, I think it was that those individuals that had lower baseline anandamide levels were more likely to derive, I mean, they're the ones that got better with CBD. And as they got better, anandamide levels in the blood rose. So Levecki pointed out, I mean, that study, if replicated, would suggest that we might be able to subtype schizophrenia based upon baseline anandamide levels. And we might be able to then offer CBD for people with a low anandamide form of schizophrenia. So from what I've read about CBD, I'm not hugely worried about it having a lot of negative problems, provided that it's actually pure CBD, not adulterated by some other stuff. And I'm cautiously optimistic that we might be able to use it for at least to some significant benefit in some people with schizophrenia. So fingers are crossed on that one.
Thanks. Yeah, I mean, and just clinically, I think most people have had the experience of their patients saying that pot is helping them, right? They're saying it reduces their anxiety, it reduces their social anxiety and depression, things like that. And there's even people who I think report it having antipsychotic properties, but that feels very anecdotal in terms of the actual.
Yeah, no, that might be the problem is between this gap between what population average or averages of study samples say and what can happen on an individual level. So I don't doubt that many people are using marijuana, like traditional marijuana, and getting benefit from it. And just like I don't doubt that people get benefit from Prozac, but with Prozac, we should know that if you have bipolar genes, you might wind up with mania. And we should know that if you take Prozac, you have about 50-50 chance of wrecking your sex life. And I'm just saying we should be upfront. People should be upfront with knowing what the risks are, and then consumers can have the benefit of choosing whether they want to risk that, or even better, if they start to notice. Like one of the things that really fascinates me about cannabis is the story with cannabinoid hyperemesis syndrome. Regular cannabis users will develop these episodes of intense and potentially life-threatening vomiting. And so hardly anybody knows about that. But if, you know, if I choose, if I'm a cannabis consumer, I feel like I ought to know that it exists so that I can correctly interpret when I wake up with nausea and belly pain and recognize it as a warning sign for something that could progress to something bad, rather than, you know, what the industry says, what that doesn't happen, or it's just a side effect of pesticide or something like that. So it's very, like, human beings are complicated and so are drugs. So I tend not to be highly critical of anybody who's telling me that they get benefit from it. I just do want them to know that it could change over time, and so here are things to look out for. I also find it useful. I mean, I find it very, very difficult to get people who are really attached to cannabis to change their use patterns. So I found it helpful to just say, I'm not trying to change your mind about stuff. You can do what you want, but you just should know that these things are possible. And you know, in my real world, occasionally people start to make these connections between 3 hospitalizations, each one preceded by heavier use of cannabis. And so the light bulb clicks for them then. I oftentimes then phrase it as, maybe we should just put it on pause for like a month and see what happens. If a person's troubled by whatever symptoms, then I can say, well, maybe let's see what happens if we just put this aside for a while. Maybe, I know it's weird, but maybe it might get somewhat better. And you know, sometimes you can get some buy-in by just not saying quit, but let's just, you know, in a spirit of curiosity, see what happens if we give it a rest. And crap, I thought I had one other thing, but those are the two primary approaches that I try to use when counseling people about it.
I want to be mindful of time. It is 9:32, so people have to go. Thank you so much for joining us today, Erik, and for Claire. I think I can, is it okay if I send your PowerPoints to folks that are asking? Okay, great.
Yeah, yeah, my PowerPoints, the last slide has some contact information, so feel free to contact me, and happy to be in touch.
Excellent. Thank you guys so much. I hope you all have a wonderful day. I will send out those slides to anybody that expresses interest. So wonderful.
All right. Thanks, everybody.
Thanks.
Bye-bye.
Take care, y'all.
Thank you.